So intermediate botany practical question paper consists 5 sections. So total marks 30, 3 hours time. So the question paper consists 5 sections. First section consists 3 families and 3 plants. Second section, that is plant anatomy. Third section, experiments. Fourth section, slides and specimen. Fifth one, record, herbarium and viva. So once again, first section, 3 families plants, that is families Fabaceae, Solanaceae, Liliaceae. Fabaceae Teprosia purpurea plant, Solanaceae Datura metal plant, Liliaceae Allium cepa plant. Second section, there will be a plant anatomy section cuttings, that is dicot stem, monocot stem, dicot root, monocot root. Third section consists of 4 experiments, that is potato osmoscope, plasmolysis, cobalt chloride paper method, and paper chromatography. Whereas fourth section consists slides and specimens, and fifth one record, herbarium and eva. So first section carry 6 marks, second section carry 6 marks, third section carry 6 marks, fourth section carry 5 marks, fifth section 7 marks, from which 5 marks for record, 2 marks for herbarium and viva versa. So intermediate botany practical syllabus, mainly it consists in alphabetical A section, B section, C section, D section, E section, F section, G section, H section. So 1A plant taxonomy, 2B anatomy, 3C live experiments, 4D vegetative morphology, E reproductive morphology, F algae and fungi, G bryophyta and pteridophyta, H gymnosperms and angiosperms. So plant taxonomy consists of 3 plants: Teprosia purpurea from Fabaceae family, Datura metal from Solanaceae family, Allium cepa from Liliaceae family. Whereas to B plant anatomy, here section cuttings are there: 4 TS of dicot stem, TS of monocot stem, TS of dicot root, TS of monocot root. Third C live experiments. The 4 number 1 osmosis by potato osmoscope, 8.2 study of plasmolysis in epidermal peel of leaf, demonstrate the transpiration by cobalt chloride paper method, separate chloroplast pigments by paper chromatography technique. Fourth D vegetative morphology, 9 storage root carrot, 10 epiphytic root vanda, 11 nodular root arachis, 12 rhizome ginger, 13 corn colocasia, 14 stem tuber potato, 15 bulb onion, 16 stem tendril passiflora, 17 thorn bougainvillea, 18 offset pistia, 19 phylloclade opensia, 20 phylloid acacia melanoxylon, 21 reproductive leaves bryophyllum, 22 trap leaves nepenthes. E. Reproductive morphology: 23 verticilluster leucas, 24 cyathium euphorbia, 25 Abenthodium ficus, 26 berry tomato, 27 Hesperidium citrus, 28 Pepocumis, 29 pome apple, 30 drupe coconut. F. Algae and fungi: 31 nostoc vegetative filament, 32 Spirogyra vegetative filament, 33 Rhizopus vegetative filament mycelium, 34 Agaricus basidiocarp. G. Bryophyta and Pteridophyta. 35 Markansia thales, 36 Funaria plant with sporophyte, 37 Pteris plant, 38 Selaginella plant. H. Gymnosperms and angiosperms. 39 Cycas microsporophyll, 40 Cycas megasporophyll, 41 Pisum P plant, 42 Gia corn plant. So from which there will be three slides are there. Nostoc vegetative filament, that is 31, 32 Spirogyra vegetative filament, 33 Rhizopus vegetative mycelium. So these are the main slides remaining from 9 to 42 specimens are there. One A. Plant taxonomy. One Fabaceae, Teprosia purpurea. Vegetative characteristics: habitat mesophyte, habit annual herb, root taproot system, stem aerial erect cylindrical herbaceous, branched, leaf colline and ramal, leaf basis pulvinous, stipulate unipinnately compound, leaf imparipinnate, alternate, petiolate, reticulate venation. Floral characteristics: inflorescence simple raceme, flower in general, bracteate, bracteolate, pedicellate, complete, bisexual, zygomorphic, perigynous, pentamerous, dichlamydous, cyclic flower. Calyx sepals 5, gamosepalous, papilionaceous, valvate. Estivation. Corolla petals 5, polypetalous, papilionaceous, estivation. Descendingly imbricate estivation. Androecium, stamens 10, didelpus 9 plus 1, anthers. Anthers are intros, dithecous, basifixed. Gynoecium, monocarpellary gynoecium, unilacular, of superior, of inferior ovary, marginal placentation, style long, stigma simple. Floral formula and identification characteristics: compound leaf with pulvinous leaf base, papilionaceous corolla, zygomorphic flower, descending imbricate estivation of corolla, didelpus stamens. Based on the above characteristics, the given plant has been identified as Teprosia purpurea of Fabaceae. Plant taxonomy. 2. Solanaceae, Datura metal. Vegetative characteristics: habitat mesophyte, wild, habit annual herb, root taproot system, stem aerial erect cylindrical herbaceous green, leaf colline ramal, simple, alternate in vegetative region but opposite in the flowering region, exstipulate, petiolate, ovate, dentate, pinnately reticulate, adenation of the petiole with the stem. Floral characteristics: inflorescence terminal, cymose, solitary cyme. Flower in general: ebracteate, bracteolate, pedicellate, complete, bisexual, actinomorphic, didelpus, heteroclamidous, hypogynous, pentamerous flower. Calyx sepals 5, gamosepalous, estivation, valvate. Corolla petals 5, white in color. Gamma petals twisted east-vation. Funnel shape. Androecium stamens 5, epipetalous, anthers intros, dithecous, basifixed. Gynoecium. Bicarpellary syncarpous gynoecium, bilacular ovary when young but tetralacular due to the formation of false septum. Ovary superior, swollen placenta, axial placentation. Ovary is oblique in position 45°. Terminal style stigma capitate. So floral formula and identification characteristics. So gametopetalous corolla, epipetalous stamens, tilting of carpel 45 degrees, swollen placenta. Based on the above characteristics, the given plant has been identified as Datura metal of Solanaceae. Plant taxonomy 3. Liliaceae, Allium cepa. Vegetative characteristics: habitat mesophyte, habit herb, root system underground bulb, stem radical, leaf simple, alternate, linear, exstipulate, parallel venation, leaf bases store the food and succulent. Floral characteristics: inflorescence simple umbel, flower bracteate, bracteolate, pedicellate, complete, bisexual, actinomorphic, trimerous, monoclinidaceous flowers. Perianth 6 steples in 2 whorls 3+3, epiphyllous, dithecous, basifixed, intros and dehiscence longitudinally. Gynoecium tricarpellary, syncarpous ovary, superior ovary, axial placentation, style terminal, stigma capitate, fruit capsule. Floral form. Identification characteristics: simple leaf, parallel venation, trimerous flowers, epiphyllous stamens, tricarpellary, syncarpous, superior ovule. Based on the above characteristics, the given plant has been identified as Allium cepa of Liliaceae family. To B. Fourth one, TS of dicot stem. Primary stem characteristics: epidermis containing stomata, conjoined collateral vascular bundles, endark xylem. Dicot stem. Trichomes are present. Cortex is well developed and differentiated into hypodermis, general cortex and endodermis. Vascular bundles are arranged in circular manner, that is U-style, arranged in a ring, that is conjoined collateral, and open vascular bundles are present in the dicot stem. Primary stem characteristics: epidermis containing stomata, conjoined collateral vascular bundles, endark xylem. Dicot stem. Trichomes are present. Cortex is well developed and differentiated into hypodermis, general cortex and endodermis. Vascular bundles are arranged in circular. Fifth one, TS of monocot stem. Stem characters: epidermis containing stomata, conjoined collateral closed vascular bundles, endark xylem. Monocot stem. Trichomes are absent. Cortex is highly radiated and represented only by hypodermis. Vascular bundles are numerous and arranged in scatterly manner, that is atactostyle. Fifth one, TS of monocot stem. Stem characters: epidermis containing stomata, conjoined collateral closed vascular bundles, endark xylem. Monocot stem. Trichomes are absent. Cortex is highly radiated and represented only by hypodermis. Vascular bundles are numerous and arranged in scatterly manner, that is atactostyle. Sixth one, TS of dicot root. Root characteristics: presence of unicellular root hairs, radial vascular bundles with alternating xylem and phloem bundles, exarch nature of xylem, tetrarch condition, small pith area is found. Sixth one, TS of dicot root. Root characteristics: presence of unicellular root hairs, radial vascular bundles with alternating xylem and phloem bundles, exarch nature of xylem, tetrarch condition, small pith area is found. Seventh one, TS of monocot root characters, presence of unicellular root hairs, radial vascular bundles with alternating xylem and phloem bundles, exarch nature of xylem, polyarch condition, that is presence of several xylem bundles, large pith area is found. Seventh one, TS of monocot root characters, presence of unicellular root hairs, radial vascular bundles with alternating xylem and phloem bundles, exarch nature of xylem, polyarch condition, that is presence of several xylem bundles, large pith area is found. Seventh one, TS of monocot root characters, presence of unicellular root hairs, radial vascular bundles with alternating xylem and phloem. Third, see 8 live experiments. 1. Potato osmoscope. So demonstrate the osmosis by potato osmoscope. Aim to demonstrate the phenomena of osmosis through uptake of water by potato tuber. Principle: plant cell absorbs water through the process of osmosis, which is the movement of solvent molecules across a selectively permeable membrane, that is plasma membrane. Observation: after the required period, it could be observed that the cavity of the potato becomes completely filled. Inference: the initial salt solution in the cavity acts as a higher concentrated solution, and in the beaker water concentration is low. Hence, water moved from beaker into potato cavity through the membrane, that is plasma membrane of potato cells. So 8.2, plasmolysis. Study of plasmolysis in epidermal peel of leaf. Aim to demonstrate the phenomena of plasmolysis in epidermal peel. Principle: the plant cells are normally in target condition due to high water content in the cell swab. When the cell is placed in hypertonic solution, the cells lose water by osmosis. Exoosmosis causes shrinkage of protoplast from cell wall. This is called plasmolysis. Observation: the wet epidermal peel is placed at the center of glass slide and pour 2 to 3 drops of salt solution on the peel during observation of peel under the microscope. The protoplast shows shrinkage due to loss of water. Inference: it is considered that the plant cells show plasmolysis when placed in hypertonic solution due to exoosmosis. Third, 8.3. Demonstrate the transpiration by cobalt chloride paper method. Aim to demonstrate the phenomenon of transpiration from leaves by cobalt chloride paper method. Principle: plant lose water in the form of water vapor. This phenomenon is called transpiration through stomata. Observation: at the end of the required period, it could be observed that the color of the cobalt chloride paper changes to light pink. This change in the color first appears as small dots here and there, and gradually as time passes becomes larger and more conspicuous regions. Inference: the original color of the dry cobalt chloride paper is light blue. When the water vapor coming out through the stomata touches the paper, it absorbs the water molecules. The blue color paper turns into light pinkish color. Third, 8, 4. Separate the chloroplast pigments by paper chromatography technique. Aim: separation and identification of leaf pigments through paper chromatography technique. Principle: the principle involved is partition chromatography based on counter current distribution. Suppose a compound A and B are two immiscible solvents taken in a container. If a compound X, which dissolves in both A and B but to different degree, is added, part of XA will dissolve and A, and in the remaining XB will dissolve in B. So the ratio XA by XB equal to AK, where K is called the partition coefficient. Observation: four differently colored horizontal zones or bands appear on the filter paper. Inference: basing on their position and color, the pigments identified as keratin, red is xanthophyll yellow, blue is chlorophyll A, blue is green, chlorophyll B dark green, respectively in the descending order. So you can see this picture: chlorophyll B, chlorophyll A, xanthophyll, keratin, and so these are the pigments which are formed on the chromatograph paper. The ratio XA by XB equal to AK, where K is called the partition coefficient. Observation: four differently colored horizontal zones or bands appear on the filter paper. Inference: basing on their position and color, the pigments identified as keratin, red is xanthophyll yellow, blue is chlorophyll A, blue is green, chlorophyll B dark green, respectively in the descending order. So you can see this picture: chlorophyll B, chlorophyll A, xanthophyll, keratin, and so these are the pigments which are formed on the chromatograph paper.
Ninth specimen, storage root, carrot. It is root modification. It is tuberous and orange in color. Taproot, stores food materials and conical in shape. Ninth specimen, storage root, carrot. It is root modification. It is tuberous and orange in color. Taproot, stores food materials. Number 10th specimen, epiphytic roots, Vanda. It is root modification, shows velamin and clinging roots. Velamin roots are long and hang in the air and absorb atmospheric moisture. Clinging roots are short, branched entire the crevices for the support. Number 11th specimen, nodular roots, Arachis. It is root modification. Rhizobium is associated with these roots. It fixes the atmospheric nitrogen. Number 11th specimen, nodular roots, Arachis. It is root modification. Rhizobium is Number 12th specimen, rhizome, Zinzer. It is underground stem modification. It grows horizontally with and dorsiventrally surfaces. It is non-green tuberous with nodes and scales. Number 12th specimen, rhizome, Zinzer. It is underground stem modification. It Number 13 specimen, corn, Colocasia. It is ground stem modification. It grows vertically in the soil. It is non-green, tuberous with nodes and scales. Number 13 specimen, corn, Colocasia. It is ground stem modification. It grows vertically in the soil. Number 14. Specimen. Stem tuber. Potato. It is underground stem modification. It is oval, tuberous, covered by brown corky layer. Eyes are present. Number 14. Specimen. Stem tuber. Potato. It is underground stem modification. It is oval. Number 15. Specimen. Bulb. Onion. It is underground stem modification. Reduced stem. Adventitious roots and found. Fleshy leaves. Bases are concentrically arranged and covered tunica. Number 15. Specimen. Number 16. Specimen. Stem tendril. Passiflora. It is a aerial stem modification. The stem is weak, leaf is simple and palmately manner. Axillary bud develops into a tendril and helps in climbing. Number 16. Specimen. Stem tendril. Pass. Number 17. Specimen. Thorns. Bougainvillea. It is aerial stem modification. It is pointed and woody structure. It is formed from axillary bud and helps for protection. Number 17. Specimen. Thorns. Bougainvillea. It is aerial stem modification. Number 18. Specimen. Offsets. Pistia. It is single internoded subaerial stem. Rosette. Form of leaves and balancing roots are present. It is found in hydrophytes and involves in vegetative propagation. Number 18. Specimen. Offset. Number 19th. Specimen. Pillowclade. Opuntia. It is aerial stem modification present in xerophytic. The stem is green noded, flat, thick and fleshy. A group of spines are present at each node. Number 19th. Specimen. Number 20. Specimen. Phalloid. Acacia melanoxylon. It is leaf modification of xerophyte. The green phalloid is formed from petiole, phalloid or alternately. Number 20. Specimen. Phalloid. Acacia melanoxylon. It is leaf modification of xerophyte. The green phalloid. Number 21. Specimen. Reproductive leaves. Bryophyllum. It is leaf modification. Epiphyllous buds lie in the notches of the margin. It carries out vegetative reproduction. Number 21. Specimen. Reproductive leaves. Bryophyllum. It is leaf modification. Epiphyllous buds lie. Number 22. Specimen. Trap leaves. Nepenthes. It is a leaf modification. It is picture shaped. Insectivorous leaves consist of digestive glands. Midrib modification into tendril. Lamina into picture and apex into lid. Number 22. Specimen. Trap. Number 23. Specimen. Verticillaster. Leucus. It is a special inflorescence. It is present between opposite leaves. Two dicaseal cymes and monocaseal scorpiid cymes are present. Number 23. Specimen. Verticillaster. Leucus. It is a special. Number 24. Specimen. Cyathium euphorbia. It is a special type of inflorescence looking like a flower. A cup-like involucus of bracts with nectar glands is seen. A single long stalked female flower and male flowers are arranged like scorpion manner. Number 24. Specimen. Cyathium. Number 25th. Specimen. Hypanthodium ficus. It is fruit-like special inflorescence. The peduncle is fleshy and cup-shaped with an opening. The female, girl and male flowers present. Number 25th. Specimen. Hypanthodium ficus. It is. Number 26th. Specimen. Berry tomato. It is a simple fleshy fruit. It has marcent calyx. The epicarp is purple colored. Meso and endocarp are fleshy with seeds. Number 26th. Specimen. Berry tomato. It is a simple fleshy fruit. It has marcent calyx. Number 27th. Specimen. Hesperadium citrus. It is a simple and fleshy fruit. Epicarp is leathery. Mesocarp is white peppery. Endocarp is divided into chambers with juicy hairs. Number 27th. Specimen. Hesperadium citrus. It is a simple. Number 28th. Specimen. Peppocucumis. It is a simple fleshy fruit. Outer part of the fruit is hard, made up of epicarp and thalamus. The mesocarp and endocarp are fleshy. Number 28. Specimen. Peppocucumis. It is a simple fleshy fruit. Outer part of the fruit. Number 29. Specimen. Pome apple. It is a simple fleshy fruit. The outer fleshy edible part is thalamus. The fruit is present inside the false fruit. Number 29. Specimen. Pome apple. It is a simple fleshy fruit. The outer fleshy edible part is thalamus. The fruit is present inside the. Number 30. Specimen. Drupe coconut. It is a fleshy fruit. Epicarp is green in color. Mesocarp is fibrous. Endocarp is hard. Endosperm is edible. Number 30. Specimen drupe coconut. It is a fleshy fruit. Epicarp is green in color. Mesocarp is number 31. Slide nostoc vegetative filament. It is a filamentous blue green algae. Filament shows chain-like arrangement of the cells which are covered by gelatinous sheath. Some cells are large, colorless, or heterocyst. They fix the nitrogen. Number 32. Slide Spirogyra vegetative filament. The filament is green branched with a row of cells. Each cell contains single nucleus with is suspended by cytoplasmic strands. Each cell contains spirally coiled ribbon, shaped chloroplast with many. Number 33. Slide Rhizopus vegetative mycelium. It shows a thallus with hyphae. Each hyphae is tubular, aseptate, and sinusoidal. It shows three types of hyphae: stolons, rhizoids, and sporangiopores. Number 33. Slide Rhizopus. Number 34. Specimen Agaricus basidiocarp. It is a fruiting body of mushroom. It shows stipe phylus. The lower side of the phylus show a hanging gills with basidia and basidiospores. Number 34. Specimen Agaricus basidiocarp. Number 35. Specimen Markantia thallus. It is a flatted dorsiventral structure. It shows dichotomous branching with apical notch at each dichotomy. Gymma cups are present on the dorsal surface, which carry out vegetative propagation. Number 36. Specimen Funaria plant with sporophyte. The plant has rhizoid stem and leaves. A sporophyte is present at the top of the branch. The sporophyte has basal foot, middle long and slender seta, and pear-shaped capsule at the top. Number 37. Specimen Terrace plant. It shows rhizome and adventitious wiry roots. Leaves are unipinnate, compound. The young leaves are spring-like and show circinate venation. The brown colored ramenta is seen on the petioles. Number 38 specimen Selaginella plant. It is a sporophyte consists of root, stem, leaves and rhizophores are present. Plant shows heterophyll, microphylla and megaphylls are present at each node. Sporophyll are formed into cone. Number 38 specimen Number 39 specimen Cycas microsporophyll. It is seen in the male cone. It is woody and triangular. The upper sterile part is called apophysis. Microsporangia in the sore or on its lower side. Number 40 specimen Cycas megasporophyll. It is found female plant and is leaf-like. It is covered by brown hairs. It has naked ovals on either side. Number 40 specimen Cycas megasporophyll. It is found female plant and is leaf. Number 41 specimen Pisum plant. Plant body is differentiated into root, stem and leaves. Taproot system, reticulate venation, pentamerous flowers are present, stipules are present, terminal leaves, leads are modified into tendrils. Number 41 specimen Pisum. Number 42 specimen Zia plant. Plant is differentiated into root, stem and leaves, fibrous roots, parallel venation and trimerous flowers are present. Silky styles and stigmas are present in spike inflorescence.